Hi, everybody. I see people logging on. Just going to wait a few minutes while people continue to log on. Looks like a lot of people today. Got a full house, a full virtual house, everybody. Just going to wait a little bit. We'll let people continue to log on, and we'll get going when the queue seems to stabilize. Well, we're about a minute after the start of the hour, and it looks like most everybody that's coming has logged on, so I'm going to begin. So hello, my name is Jeff Smith, and I'm the Strategic Partnership and Outreach Specialist for the Institute of Cannabis Research, hosted at Colorado State University, Pueblo. Welcome to the Cannabis Research Webinar Series, which is presented by the ICR and the Lambert Center for the Study of Medicinal Cannabis and Hemp at Thomas Jefferson University. This webinar series, in collaboration with the Lambert Center, is one of the many ways that the ICR works to promote the dissemination of cannabis research. For example, please see our website, which can be found by Googling the words Institute of Cannabis Research, and navigate to our Outreach tab, and you can find our current e-newsletter. Or you can go to the Research tab and you can review summaries of each of the 20 major research projects that the ICR currently funds. You can also find information about future monthly webinars and review past ones on the website. We anticipate our new website will launch in the very near future, so please stay tuned. You can use our current website to sign up on our listserv for updates. Additionally, please note the link to the Journal of Cannabis Research, which the ICR sponsors. The journal is published by prestigious Springer Nature and is always looking for both high-quality scientific article submissions and reviewers. Additional information about the ICR is also available on our website, so please visit us. Before we start today, I would like to mention some logistical items for the webinar. Today's presentation will be followed by a short question and answer period. To ask a question, please use the Q&A feature in the Zoom toolbar. You can enter questions at any time during the presentation, and we will address as many questions as time allows at the end of the hour. Please reserve the chat function in Zoom for technical issues and questions related to technical issues. Both the Q&A and the chat functions can be found in the toolbar on your Zoom screen. It's now my pleasure to introduce my colleague, Ruth Charbonneau, the Associate Director of the Lambert Center, who will be introducing today's presenter and moderating the webinar.
Thank you, Jeff. We're pleased today to have Dr. Emily Bates as our speaker. She is an associate professor in the Department of Pediatrics at the University of Colorado School of Medicine. Dr. Bates studied neurodegenerative and ion channel function as a PhD student at Harvard Medical School and conducted research about underlying mechanisms for neurological disorders as a postdoc at the University of California, San Francisco School of Medicine. She started her laboratory at the University of Colorado in 2013. Her lab focuses on how ion channels impact embryonic and fetal development. Regulation of ion channels is also altered by components of cannabis, such as THC and cannabidiol. CBD crosses the placenta, and many of the ion channel's CBD effects are expressed in the developing brain, suggesting that maternal CBD consumption during pregnancy could alter offspring brain development. There was little research about how CBD affected fetal development to indicate whether it was safe. So Dr. Bates' research on the importance of ion channels in development, she felt that there was an urgent need for research defining the consequences of CBD exposure on offspring development. Funding from the Institute of Cannabis Research enabled her and Karli Swenson to perform important experiments to discover that oral CBD consumption during pregnancy increases the perception of pain, decreases cognitive ability, and decreases activity of the cortex in mouse offspring. Today, she'll be presenting on is CBD consumption during pregnancy safe, postnatal consequences of CBD exposure during gestation. And with that, I'll turn it over to you, Dr. Bates.
Thank you so much, Ruth and Jeff, for that kind introduction and for inviting me to give this talk today. I'm excited to talk with you. Okay. Excuse me. So people use cannabis products to treat nausea, anxiety, and pain. And these are all symptoms that are very common in the pregnant population. While only 2.5% of pregnant people report cannabis consumption to physicians during pregnancy, about 22% of cord blood samples in pregnant people test positive for THC, indicating that there is cannabis use in 22% of pregnant people here. Excuse me, clinical studies show gestational cannabis exposure does have some negative consequences, including reducing birth weight and increasing the number of pregnancies that have preterm delivery. It reduces the size for gestational age and increases admissions to the neonatal intensive care unit. It also is associated with increases in adiposity and fasted blood glucose levels at age 5 for children that were exposed to cannabis in utero. And it has an association with increases in anxiety and attention deficit and hyperactivity disorder. However, cannabis is composed of multiple components, and the primary cannabinoids that are found in cannabis are THC and CBD. And while THC is legal here in Colorado, it's not federally legal. It's psychoactive and it's a Schedule I drug for the federal government. And so public perception really varies by region and culture. Here in Colorado, it's very widely accepted, but just in our neighboring states, it's less accepted, right? However, cannabidiol or CBD is non-psychoactive and it is federally legal, and it's widely socially accepted in the United States in general. It's really marketed more as a health supplement, and so people perceive it to be safe. But just like THC, CBD that's consumed by someone, a pregnant person, will go into the bloodstream and it will cross the placenta and enter fetal tissues. And it can be found in both the fetal brain and in other fatty tissues such as the liver. So an increasing population consumes CBD during pregnancy because they believe it to be safe. But we would really like to know if CBD contributes to these adverse effects of fetal cannabis exposure. To test these ideas, animal studies really have advantages in that we can give defined, consistent doses and control the route of administration. We can also control the timing of exposure and distinguish between different components of cannabis. We can also control for the effects of other substances by not including them in the diet of the mouse, whereas in a clinical population, we have no control over diet or other drugs that could be consumed. So how might CBD exposure impact fetal development? It turns out that CBD impacts the activity of several different ion channels and G protein coupled receptors, and even transcription factors or nuclear receptors such as PPAR gamma. So I'm just going to walk you through some of these. So CBD activates an ion channel, a calcium channel called TRPV1. This calcium channel is primarily known for its ability to sense thermal stimuli. It's expressed in the fetal hypothalamus, and it also regulates pancreas function. It's been implicated in anxiety behaviors as well. And then CB1 and CB2, we think of really commonly when we think about the effects of cannabis, but CBD does not actually directly bind CB1 or CB2, but it can have activity on these G-coupled receptors through its effect on increasing a ligand that's expressed, the natural endogenous ligand of CB1 and CB2. It also can negatively modulate these G-coupled receptors as well. CBD activates 5-HT1A, which is a serotonin receptor, which is implicated in cognitive function and is expressed in the prefrontal cortex during development. And then CBD also activates, actually antagonizes GPR55 and activates, has inverse agonism, which actually means that it inhibits GPR3, 6, and 12. So it has negative effects on these G protein coupled receptors. And then it activates PPAR gamma, which is a fatty acid sensor that regulates metabolism, and is expressed in the fetus as well. So all of these receptors that are shown here are expressed in fetal tissues, and can be affected by CBD consumption during pregnancy. So this led us to believe that CBD exposure could affect brain development and postnatal behavior, and this needed to be tested. And so these experiments were started by Karli Swenson, who was a graduate student in my lab. And so what she did was she administered 50 milligrams per kilogram CBD to wild type or TRPV1 knockout animals by oral gavage daily from embryonic day 5 through birth. And then at birth, we took some of a subset of these animals and performed a technique called single-cell RNA sequencing to look at gene expression in the hypothalamus, which is a region in the brain. And then the rest of the animals were allowed to grow up, were weaned to normal chow, and then underwent a whole battery of behavioral tests, including the open field test, the Y maze, the puzzle box, and thermal sensitivity tests. And then we looked at metabolism later on. And so there was these animals went through a whole slew of behavioral tests. But first, it's important to note, did the CBD actually get to those pups, right? And so we looked at the levels of CBD and its metabolites in the dam and the pup plasma and found that we could see CBD and its metabolites 2 hours after a dose at embryonic day 18.5 and at P0, but it was really negligible by postnatal day 4 and beyond. This graph also shows that the levels of CBD that are found in the pup plasma are really very similar to those that are found in the dam plasma, telling us that the placenta is not providing any sort of barrier for CBD to reach the pups. In addition to this, it's also important to note that we don't see CBD or its metabolites after P4, which tells us that any effects that we see beyond that time point are because of developmental changes that have occurred in the pups and not because of acute effects of CBD. So Karli went through, looked and cataloged all of the factors that could potentially affect the development of these animals kind of as a correlation. So she looked at the total number of pups per litter, the alive pups at weaning, so survival of those pups, and the average pup weight as determined by dividing gestational weight gain by the number of pups per litter, and found that there were no differences between those that had been exposed to CBD or vehicle in any of these measures. She also looked at gestational length, gestational weight gain, and the sex of the offspring, and none of these were affected either by the treatment group. So the first test that I performed actually was to look at how CBD exposure might affect pain sensitivity, thermal pain sensitivity. And this was because TRPV1 is that receptor that is activated by thermal stimuli. And so it regulates thermal pain circuits. And so the fact that it could be excessively activated during pregnancy could alter those thermal pain circuits during development. And so this is called a Hargreaves apparatus, and the mice sit on a glass plate, and then we shine a little light under the hind paw, and it warms up at a consistent rate. And so then we measure the latency to response to that thermal stimuli. And those that respond faster are more sensitive to thermal pain. So what we found here was that female animals that were exposed to CBD or vehicle were not significantly different from each other, dependent on the treatment group. And even that was the case even when we controlled for estrous cycle, which can sometimes change thermal stimuli. However, males that were exposed to CBD during gestation were significantly faster to respond to that thermal stimuli, telling us that they had an increase in perception of thermal pain compared to the vehicle-exposed animals. And now this was not the case when these animals did not have a functional TRPV1 channel. So that means that this alteration in the circuit that is sensing thermal pain depends on the activation of TRPV1. The effect of CBD on that developmental circuit happens through TRPV1. Okay, now Karli really wanted to test whether CBD exposure during gestation would have any effect on anxiety, and this was rationed because her rationale for that was that people who are exposed to cannabis, whole cannabis, throughout gestation or during gestation, really have an increase in anxiety behaviors. And so she tested this in 3 different ways. She really tested this quite thoroughly, looking at using the open field test, the light dark box, and the elevated zero maze. At 3 different time points that roughly correspond to a mouse's puberty. So this is Karli right here in the corner. And so using this, the open field test, I just wanted to show you what this looks like. These right here are little mice, and a computer tracking system is filming them and then tracking where these mice go. And it takes note of how much time they spend in the middle compared to the sides. And mice that spend more time in the middle are less anxious, and those that spend more time on the sides those that spend time in the sides are less anxious, are more anxious. Sorry, I keep getting that mixed up. Okay, and so long and short of it is that through all of these measures, Karli found that the mice that were exposed to CBD, either female or male mice that were exposed to CBD throughout gestation, had no difference in anxiety, but through any measure compared to vehicle-exposed counterparts. So this tells us through the open field test, the light dark box, and the open field and the elevated zero maze, that there was no difference in anxiety measures in animals that were exposed to CBD compared to vehicle throughout gestation. Now, the next question was if CBD exposure impacts cognition. And the reason that we thought that this might be the case was because those serotonin receptors that CBD activates do regulate cognitive function. And so we thought that this potentially could have an effect on cognition. So these animals are placed, this is called the puzzle box, and these little animals are placed in this puzzle box, and then they want to make it to this goal zone, which is really a dark box that's more comfortable for them. And so we make this more and more challenging by covering doorway and making it more and more difficult for them to figure out how to get to the goal zone. And then the time to reach the goal zone is measured. So here you can see from the females that there's not a significant difference when we give them an easy challenge. But once we give them a hard challenge, the animals that were exposed to CBD compared to the vehicles took a lot longer to reach that goal zone than did their vehicle exposed counterparts. And so this tells us that in the very most difficult challenges, that CBD exposure impaired cognition or problem solving ability. But this was not the case for the males. So as you can see, the males found there was very little difference between the males' performance on this test. And so when we found this, we thought, well, this might be because of how the 5-HT1A receptor is implicated in cognition in the prefrontal cortex. And so we collaborated with Dr. Wanchan Oh and Luis Gomez-Wolschner, who is his student, and they work just upstairs from us and are experts in measuring electrophysiology and morphology of neurons in the prefrontal cortex. And so in collaboration with them, we dosed these animals, wild-type animals, with 50 milligrams per kilogram CBD throughout gestation from embryonic day 5 through birth, and that at postnatal day 14 through 22, Dr. O and Luis performed 2-photon imaging, electrophysiology, and glutamate end caging, and I'll walk you through these experiments. So they fill these neurons with dye so that they can see them, and then they image them. And then through that, we can analyze the size and shape and number of spines in the dendrites. And Luis and Wanchan and Tori worked together to analyze these and found that there were no differences in spine density or morphology or size between CBD exposed and control exposed animals. They then went on to test electrophysiological properties of these neurons. These are layer 2-3 neurons in the prefrontal cortex. And they found no difference in resting membrane potential in either the male or the female animals. However, they found that the minimum current to action potential was significantly increased in CBD exposed animals compared to vehicle exposed animals. This difference in the amount of current that was required to elicit an action potential was due to the very significant difference between the females that were exposed to CBD compared to vehicles, because the males that were exposed to CBD were not significantly different from vehicle exposed animals. The amplitude of these action potentials was, the amplitude that was required for an action potential to fire was significantly higher in the CBD exposed compared to vehicle exposed animals. And again, this was because of the significant difference between the CBD exposed females compared to their vehicle exposed counterparts. We did not see a significant difference in the male mice. Now, also, the number of action potentials at any given current was significantly reduced in the CBD exposed animals compared to vehicle exposed. So here we get a similar current, and here you can see that there are only 3 compared to the 6 action potentials here in the vehicle exposed. And this is quantified at multiple different current injections. And you can see that, again, the males show an insignificant difference, whereas the females are significantly reduced compared to vehicle exposed animals. So then the next question was whether all of these measures that I'm showing you before are kind of presynaptic measures, but we can also look at postsynaptic measures by uncaging glutamate with a chemical cage that's activated by or opened by light, really. So you use 2-photon, very laser focused on this glutamate cage. It opens, and that allows the glutamate to reach his spine. It's a very controlled amount of glutamate, and then we look at how much response you get from the postsynaptically from that glutamate uncaging. And you can see here in the females that there's a lower amplitude of evoked potential from the females that were exposed to CBD compared to the vehicles, and that was not the case in the males. Now, these spines are very similar in size. They're size matched, and the animals are age matched. So really the only difference is the exposure to either CBD or vehicle. So here for this first part, fetal CBD exposure really increases thermal pain sensitivity in male animals, but it does not affect anxiety behavior. I didn't tell you this data, but it does not impact compulsivity or spatial memory in our tests, but it does decrease female cognition. And it really reduces the electrical activity or the excitability of the prefrontal cortex. And we wonder if this might be due to excessive activation of a serotonin receptor 5-HT1A. And so we still have some remaining questions that we're going at, which is, what is the mechanism underlying this CBD-induced cognitive impairment? Is it dependent on 5-HT1A or one of the many other receptors that CBD affects? And we're really interested in the mechanism for sex differences in response to fetal CBD exposure. Now, I told you at the beginning that we also took hypothalamus from newborn mice after the CBD or vehicle exposure, and we did a technique called single-cell RNA sequencing, which means that these hypothalamic cells are dissociated into single cells. Each of those cells is lysed, and the genes that are expressed are given an independent, unique barcode. So we can identify which genes are expressed in one individual cell. And then those cells can be grouped together based on how similar their gene expression is. So we can really identify different nuclei within the hypothalamus to figure out what type of cell they are and how their gene expression is changed within that single cell. And so these plots, this gives us something called a UMAP. This was done with the help of Becky O'Rourke, who is a bioinformatics specialist here at the University of Colorado in the section of developmental biology. So she helped us put together this UMAP. And so these here in the center, these are neurons, and these are glial cells and OPCs. And so you can see we can actually identify within these populations of cells that are important for hypothalamic function. And so within these specific hypothalamic nuclei, we found that CBD exposure changes gene expression. And then we can group those genetic differences in gene expression into processes that they would affect. And those changes in gene expression predict that we would find differences in brain development, like general brain development, in axon and dendrite genesis, so the making and growing of axons and dendrites, and also in synaptogenesis, so the formation of synapses. So this told us that there are likely candidates that we can follow up that will explain some of the differences in behavior that we've seen in these animals. But something that was surprising to us and exciting was that we also found that in some centers of the hypothalamus that are responsible for feeding and exercise behavior, or otherwise known as energy balance behaviors, there were differences in gene expression that predict reduction in insulin secretion and in insulin sensing as well. And so we started a new kind of experiments with these mice, looking at energy balance behavior and body composition and insulin sensitivity. So the question is, does fetal CBD exposure affect offspring hypothalamus development? And how does that change energy balance behaviors, body composition, and insulin sensitivity? So the hypothalamus is a part of the brain that's really important for regulating exercise, feeding behavior, and metabolism in general. And so within some of these centers of the brain, the hypothalamus, that are responsible for energy sensing and energy balance, we found that POMC was decreased in expression. POMC is important for leptin and insulin sensing to reduce eating when we should be full and we should be done. So a reduction in POMC expression is associated with dysregulation of energy balance, overeating, and severe obesity. We also found that CBD exposure increases the expression of PDE10A, and PDNA is important for also regulation of energy balance, and increases in expression of PDE10A are associated with overeating with exposure to high-fat diet. So you might not eat too much when you're just presented with a salad, for example, but when there's a cheesecake around, you're gonna go for it, right? If you have too much PDE10 expression. And this is also associated with increases in adiposity. Now, these mice that we have, that I'm talking about right now, we'd already weaned them to a normal chow, and so they were basically on, a healthy diet. But we still wanted to test whether there were differences in eating behavior and energy expenditure, these things that feed into metabolism and energy balance. And so we collaborated with the Nutrition Obesity Research Center here at the University of Colorado. This is called the NORC, and they performed these experiments where they put our mice into a very calibrated box that measures their energy expenditure and their how much they eat and their respiration, really, which is an indirect measure of metabolic rate. And what they found was that there was no difference in energy intake, that means eating, total energy expenditure, or activity like exercise or physical activity level, or other measures of metabolism in the female mice or the male mice. We found no differences in these animals. Now, just to remind you, these animals were fed on a normal chow diet, so a healthy diet, basically. And so we really wonder if we expose them to a high-fat diet, if they would be more susceptible to the effects of high-fat diet than animals that were not exposed to CBD or were exposed to vehicle. So, not surprisingly, when we found no differences in energy balance behaviors, we also found no difference in body composition as measured by weight, lean mass, or fat mass, or when we do the ratios and look at percent lean mass or percent fat mass. And this was measured by MRI. So then the next question is, does fetal CBD exposure impact glucose tolerance and insulin sensitivity, which are the hallmarks of diabetes? So to test these, to perform these types of tests, we put animals through what's called a glucose tolerance test. Now, if any of you have been pregnant, you'll have experienced the joys of a glucose tolerance test. And this is when we fast over a period of time. And so we fast the mice over 12 hours for these experiments. And then we give them a bolus of, we test their blood glucose at that fasted state, and then we give them a bolus of glucose. When you take a glucose tolerance test as a human, it's a drink of very sugary water that tastes disgusting. And then over time, we look at the blood sugar levels in response to that glucose that's taken in. And so for these mice, you can see that in the beginning at fasted state, you can see where their blood sugar levels are. Then we gave them a bolus of glucose, and their blood sugar goes up, as you can see here. Now, those that are in the blue line are those that were exposed to CBD, and you can see that that blue line goes up much higher than it does in the black line, which is the control animals, the vehicle exposed animals. So this tells us that these animals can't deal with the glucose as well when they've been exposed to CBD throughout gestation as their vehicle exposed counterparts. So this is called that they have impaired glucose tolerance, where they're glucose intolerant. Now, this was the case for the male animals that were exposed to CBD compared to controls. But in the females that were exposed to this high dose of CBD, we didn't see a significant difference. However, when we looked at lower doses of CBD exposure throughout gestation, so the same paradigm, same oral consumption, but 10 milligrams per kilogram and 5 milligrams per kilogram, we still see glucose intolerance in the males that were exposed to 10 milligrams per kilogram CBD exposure. But for the females, we actually found glucose intolerance at both the lower doses, both the 5 milligrams per kilogram and the 10 milligrams per kilogram CBD exposure. So both in males and in females, we see impaired glucose tolerance with CBD exposure throughout gestation. Now, remember, these animals don't have any difference in behavior as far as feeding or energy expenditure, but they're becoming more susceptible to diabetes without those differences in behavior or body composition. And so why could that be? It could be that, so glucose homeostasis is really regulated by the pancreas, right? And also the response of the responsive tissues. So when we take in glucose or we eat a big meal, our pancreas senses that that increase in blood sugar. And so the islets, pancreatic islets, secrete insulin in response to that blood sugar. And that insulin tells muscle and fat and the liver to take in the glucose and then store it in their respective ways as glycogen or as fat. So the first test is, are those islets that are supposed to be secreting insulin, are they formed properly when they've been exposed to CBD throughout gestation? And so Madison Rodriguez, a graduate student in my lab, and Karli Swenson, a former graduate student in my lab, made sections of pancreas tissue from vehicle exposed and CBD exposed female and male mice, and quantified the number of cells in those islets, and then the number of them that are the type of cells that secrete insulin, called beta cells, or alpha cells, which are cells that secrete glucagon. And what they found was that there was a trend towards a decrease in the total number of islet cells in an islet, and that was because of a decrease in the number of beta cells in an islet. But there was no difference in the alpha cells per islet in the 50 milligram per kilogram CBD exposed animals. However, they did not find that difference in the 10 milligram per kilogram exposed animals. So there's a slight reduction in number of beta cells, those cells that secrete insulin, but do those cells function properly? To test this, we worked with Dylan Scarborough, who is in Dr. Lori Cecil's lab here at the University of Colorado in the Diabetes Research Center. And what we did here was collect blood, again, fast animals, and then test their fasted blood glucose levels and their fasted insulin levels. And then give them a bolus of glucose and look 5 minutes later, and we should see an increase in the levels of insulin in the plasma in response to that glucose. That's how a pancreas is supposed to work. However, here we see that while the fasted insulin levels are the same in the CBD and vehicle exposed animals, after we give them a bolus of glucose, the vehicle exposed animals increase their levels in insulin levels. However, the CBD exposed animals do not increase their insulin levels significantly. This tells us that the pancreas is not working properly. And this occurs at both the 50 milligrams per kilogram dose and the 10 milligrams per kilogram dose of CBD. Excuse me. Okay, so now we know that the animals that have been exposed to CBD, either a high dose or a medium dose of CBD throughout gestation, this impairs pancreas function over time, even in adult animals. Then the next question is, what about those tissues that are supposed to be taking in the insulin, in the glucose? Are they responding to insulin adequately? And so to test this, we use what's called an insulin tolerance test. So these animals, again, are fasted for just 5 hours, and then they are given, we test their blood glucose levels, we give them a bolus of a dose of insulin, and then we look at their blood sugar over time, and that should go down if they're responding to that insulin injection. So even at low doses, like 5 milligrams per kilogram of CBD, the lowest dose that we test, really, it's not such a low dose, but the lowest dose that we tested of CBD, at 5 milligrams per kilogram, we see insulin resistance. See, as this blue line, that's the CBD exposed animals, are really quite resistant to the effects of that insulin compared to the control animals. And the same was true for the female animals. So both female and male animals are insulin resistant. So the conclusions here are that CBD exposure really impacts the development of energy balance circuits in the hypothalamus as measured by gene expression. And CBD-induced changes to gene expression predict increased susceptibility to high-fat diet-induced glucose intolerance and increased adiposity, dependent on that high fat. We need to test this hypothesis. This is something that we really want to do, and we're hoping to get funding to do. We also found that CBD-induced energy balance behavior and metabolism changes in young adult mice are really subtle, and they don't result in any changes in body composition. However, we found that fetal CBD exposure induces glucose intolerance in both male and female offspring, and that it also induces insulin resistance in both male and female offspring. So we really, in the future, would love to know if CBD exposure has direct effects on pancreatic islet insulin secretion, and if this high-fat diet, if high-fat diet exacerbates CBD-induced glucose intolerance, and if CBD-exposed offspring are more susceptible to high-fat diet-induced adiposity. We're also interested in what the mechanism is that explains how fetal CBD exposure impacts glucose tolerance and hypothalamus development. And with that, I'll thank you for your attention and give credit to those who should get credit here. So Karli Swenson really started this project, and I gave her credit for what she did throughout the presentation. Madison Rodriguez has taken over the project, trying to understand how CBD exposure affects the pancreas and pancreas development. And the other people in my lab, former members of the lab who contributed to this project, are Cameron Korth and Lillian Foltz, who and both Karli and Lillian were recipients of the Institute of Cannabis Developing Scholars or Scholars Awards. And so we're really grateful for that funding that we've received from the Institute of Cannabis Research. And then Dr. Kristen Boyle's lab is helping us really tease apart the molecular mechanisms and cellular mechanisms for the insulin resistance we've seen. And we've also received help with this from members of her lab, including Vincent, Manuel, and Ali. We're very grateful to the NORC and help from Dr. Paul McLean and Matthew Jackman. Dr. Lori Cecil has consulted with us throughout this project, along with Dylan Scarborough. And then, of course, Wan Chan O and his lab has been really a valuable resource in teasing apart the mechanisms underlying the cognitive impairment that we observed. And of course, we're thankful for funding from the Institute of Cannabis Research, the Diabetes Research Center, NIH, and NSF, and the Colorado Clinical and Translational Sciences Institute. So with that, I'll take any questions.
Thank you, Dr. Bates. I want to start with a question from one of your early slides about the presence of cannabis in the cord blood. And just, I think you said 22%. Was that really just to look at the veracity of the reporter, or did it tell you anything about sort of potential timing of when cannabis may have been used?
Oh, so that was not data from us. That was from Dr. Erica Wymore's lab, I believe. And so that was clinical measures, not from our lab, but they were looking at just whether it was there, THC was there, and they weren't not looking at CBD. So I'm really interested in whether if we looked at CBD rather than THC, we would see even more exposures because of the CBD being sold separately, right? And there are many people who would never touch whole cannabis, but would readily take CBD for one of these variety of symptoms that they experience during pregnancy. So yeah, but the CBD studies have not been done yet.
Okay. So how consistent across mice studies examining prenatal CBD and or THC exposure that there are no differences in birth outcomes between the treatment and control groups?
Birth outcome meaning, so from the CBD exposures, the labs that have published on that and those that I've talked to independently have said that they don't see any differences in gestational weight gain, gestational length, and gestational the number of pups, for example. None of those measures seem to be different between labs that have done this, and we didn't see any differences at any of the doses that we tested. For CBD. For THC, that is not the case. And there are differences there in some of those gestational effects. Yes.
Next is, what type of vehicles were used for males and female rats?
Mice. Yes, so we used sunflower oil to dissolve the CBD in, and so sunflower oil was given to the vehicle exposed mice.
A comment, that's super interesting data, and thank you for sharing. At what anatomical site do you think the decreased insulin sensitivity is occurring? Liver, skeletal muscle, adipose? And a second part, is there an influence of fetal CBD exposure on glucagon secretion from the alpha cells?
Oh, those are great questions. Those are actually questions that we're pursuing actively. So what we have done so far with the help of Dr. Kristen Boyle's lab is look at insulin signaling proteins downstream of insulin signaling. And we have not yet found a tissue that we think is responsible. But because of the numbers that we had to make those measurements and the way that they were collected, we're not confident in that yet. So we do not have evidence of one particular tissue being responsible. However, CBD target proteins and receptors that are activated are expressed in each of those tissues. So in fat, in liver, and in muscle. So TRPV1 is expressed in all three of those and has a role in metabolism in fat, for example, and in glucose and in insulin secretion, for example. And then some of the GPRs are expressed in each of those tissues as well. And so right now the jury is out, but we would love to know that, and that is one of the reasons why we're applying for another ICR grant to try and figure that out. But it costs a lot of money to do these experiments, so we really we will do those experiments when we have the funding to do so. Yeah.
How do the doses studied in this research compare to doses that people typically use?
Okay, great question. Yeah, so we started with a very high dose, one that no one will ever take. So 50 milligrams per kilogram is above what anyone would take, a person would take. And we did this to uncover any potentially subtle effects that might be difficult to see in the numbers that we were looking. And so we started with a high dose, 50 milligrams per kilogram. Now 10 milligrams per kilogram is the dose that's prescribed for Epidiolex, which is a CBD product that is FDA approved to treat epilepsy. And so that dose is one that is consumed by humans. And then 5 milligrams per kilogram might be a dose that people take over the counter, for example. And now the metabolism of that drug will be different in a mouse than it is in a human. And so it is important to look at a range of doses, starting with high and then going to a lower dose.
Would you expect the expression levels of the receptors that bind to CBD to be permanently impacted in the postnatal pup that is no longer experiencing CBD exposure? And a second part, or does the expression of these receptors restabilize once CBD exposure is limited?
This is a great question, right? And so we think that excessive activation of a receptor, for example, or inhibition of a receptor is making physical changes to the cells that it's affecting, right? So for example, when TRPV1 is excessively activated, what we might expect and what we think might be happening is for that increase in thermal pain sensitivity, that there's an increase in TRPV1 expression in the cells that already expressed TRPV1. And this may be potentially because TRPV1 is excessively activated, but whenever it's activated, it's also inactivated directly afterwards. And so it could be that the cell increases expression of TRPV1 in response to that excessive activation and then inhibition of the channel or inactivation of the channel. And so we think that there probably is a difference in the expression of the receptors themselves. But in addition to that, there can also be cellular consequences to an excessive activation of a channel. For example, many of the receptors that TRPV1 activates excessively or inhibits affect axon guidance. Even CB1, for example, and GPR55 are important for axon guidance and axon growth. And so the inhibition of GPR55 or the potentially excessive activation or inhibition of a CB1 could potentially affect axon guidance. And then that axon is not the right length, does not reach the right targets, and could therefore affect really the neuronal networks, the circuits that are being formed. So we think that there's actually there could potentially be cellular consequences in addition to differences in gene expression that we see. And then in addition to that, there are transcriptional differences, right, that we see in the hypothalamus, which could change the identity of cells and also their function long term.
Thank you. This is a slightly different direction, but it was included in your bio. So your personal experience in terms of your presentation here and the need for more research, how does that translate to prescribers or to dispensaries where they're sort of recommending products to women who are pregnant?
Right. I think that's the reason that we need to do this type of work, and we need to have communication with people. And this is one of the reasons why these types of seminars are so important, right? So that there's a dissemination of the information that we as researchers obtain. So yes, my experience was not even with dispensaries. I would just be at the grocery store, and because I was nauseated, and it was very obvious that I was nauseated, people from friends to strangers would recommend that I use cannabis products to treat that nausea. And if I didn't do research on how ion channels affect fetal development, I would have been, I think I would think, well, this would be good. I don't like being nauseated, right? So I have a lot of sympathy for people who just the general public wouldn't have any reason to think that there should be an effect of CBD on a developing baby that they're carrying. And so it is really important that we do this type of research, right? This is really something that everyone wants to do the best thing they can for their baby. There's not a person that would intentionally harm the baby that they carry. And so, but at the same time, you want to take care of the mother, make sure that she's happy and healthy. And so if the hope was that no matter what we did with these experiments, we would find something that would help the world, right? Either that there were no negative consequences of taking CBD during pregnancy, and then it could be used as a nausea treatment or something to calm you down and help you go to sleep at night when you're pregnant. But that was not the case in the experiments that we performed. We found that while there was no effect on anxiety, there were effects on pain sensitivity, and there were effects on these on cognitive function, and then also on this kind of susceptibility to diabetes. Yeah.
Okay. I would also say the sort of possible long-term effects as well that are still unknown for not just initial development. So.
Right. So all the experiments that we did were in adult mice, and besides the single cell RNA sequencing, or kind of on the verge of adult mice. So we've actually, so I think you're right there. Yeah.
And just sort of any thoughts in terms of the gender differences that you found? Is that consistent with other studies, or sort of any sort of thoughts in terms of that finding?
Right. Yeah. So there is another study, Warren et al., where mice were given oral CBD also. They were given a lower dose. So they started with 20 milligrams per kilogram. So a different dose than we were giving. And they dosed animals prior to copulation, and then also throughout all of lactation as well. So they did that oral gavage for that whole time. And they also found differences in behavior that were sex specific. They only found differences in the female mice and not the male mice. The differences that they found were in actually spatial memory and compulsivity as measured by the marble burying. Now, we also performed marble burying tests on our mice, and we did not find the same differences that they found. But that could be a difference in the dose or the duration of the exposure. But theirs were also sex specific. So I think there is something there. The mechanism is hard to figure out. It's hard to figure out. There are higher levels of circulating serotonin in females than males. So maybe it's that you're excessively activating and going over a threshold or something like that in the females that you don't reach for the males. Or maybe there's, I don't know.
Yeah.
There's a lot of different ways that my brain goes in that, but we don't have evidence for any of them at this point. So it's right now all hand waving.
Well, thank you. I really enjoyed the presentation and questions from the audience. We appreciate you taking the time to be with us today. And as mentioned, this will be up on the ICR website for recording. And if anyone has any questions, may they reach out to you, Dr. Bates?
Yes, please. Yes.
Great.
Yeah,
all right, well, thank you. And I just would ask everyone to join us for next month's webinar, which will be sent out by sort of the newsletter and notices. So thank you for joining. And thank you, Dr. Bates.
Oops. Okay.